going to sort of talk a bit about the elephant in the room. And that is the concerns, the worries about relapse of our lymphoma, and use that as a means of discussing clinical trials that we have available in Australia in a little bit more depth. Because for those of us who are newly diagnosed with lymphoma, who are very hopeful of either a cure or a prolonged remission, or those of us here who have had 1, 2, 3, 5 or however many relapses, I think it's very important that while this is a very confronting concept, that everyone gets, I hope everyone gets the take-home message today that relapsed lymphoma is still very treatable with so many of the promising new therapies that Professor Saal has introduced us to, as well as bendamustine. Just coming back to some of the terminology that's already slipped in, and to just explain a little bit of that to you. We doctors, we say the disease is refractory to our therapy when it's growing during the chemotherapy. Now that's actually very rare in the first line setting, but it becomes increasingly common as people have second or third or subsequent therapies. When your doctor tells you your disease is stable, effectively that's translation for it's less than 50%. There's been a less than 50% reduction in the volume of the lymphoma, but we haven't yet obtained a partial response whereby we've had a 50% reduction in its volume on the CT scan, but there's still some residual disease that we can see on the CT scan. A complete remission is defined traditionally when on a CT scan we can't see any enlarged nodes present which reflect the presence of the lymphoma. And maybe later on in the question and answer session, on the couch session, we can discuss the actual definitions of complete remission, because certainly as Professor Saal has explained, PET-CT scanning is really replacing CT scan as the gold standard for definition of complete remission for most lymphomas. But in addition, with greater sophisticated detection of lymphoma residing in the body, we often also have some very sensitive blood and bone marrow tests which help us to define complete remission for some lymphomas. Beyond achieving remission, it's really only Dr. Time who determines whether you are cured. Professor Rimmel talked about how we can give you prognoses. We can talk to you about the average. We can talk to you about the highest 10%, the lowest 10%. But in the end, all that we can do is give you our best estimate to help you arrange, your own, social life planning. And that's very important, and please don't get disheartened or hopefully be very hopeful when you manage to prove your doctor's predictions very wrong. Now, relapse is defined as disease that's come back after you've obtained a complete remission, whereas progressive disease is disease after you've either been in a stable or a partial remission. Now, relapsed lymphoma, as I said, is still very treatable, particularly if the relapse has occurred after being in remission for several years, and if the patient still, regardless of what age they are, if they are still very fit and capable of receiving additional rounds, if you're willing to, and willing indeed, to go it again with some further treatments. And second and even third, more remissions can last for many months. And indeed, for some lymphomas, even after a first relapse, the goal of treatment is still cure. But I want to talk you through in a bit of detail the treatments that we now have in 2005, which are very much based on the huge advances made through clinical trials research in the last 20 years in particular. And the reason I've focused on relapsed lymphoma is because that is generally where new therapeutic treatment approaches, new therapies themselves are introduced into the practice of lymphoma care before they start moving up into being introduced in the frontline setting. But certainly, because of the promise of so many of these new therapies, a number of you are actually receiving these new therapies in the context of your first line of therapy rather than for multiple year relapse disease. And that makes it a very exciting time for us, because our research is certainly moving us away from absolute vodka, the very blunderbuss, very toxic chemotherapies that we had in the 70s and 80s, onto these smarter targeted therapies. Rituximab, the MabThera, the anti-CD20 antibody, is no longer a novel new research agent. It's very much part of the standard, whether it's combined with bendamustine, whether it's combined with CHOP, whether it's combined with anything else. It's very much a standard of care these days. But Professor Sahls talked about the GA101 or obinutuzumab, the anti-CD20 antibody, that appears to be more powerful in treatment of some lymphomas in early clinical trial results, which need further maturation before we can really give the green light to the use of obinutuzumab. Similarly, brentuximab is targeted against the protein CD30 present on Hodgkin lymphoma cells, as well as many of the the rare T-cell lymphomas. And brentuximab delivers that toxin that Professor Sahls was talking about right to the lymphoma cell. We also have access through clinical trials now, the ibrutinib and other Bruton tyrosine kinase inhibitors that inhibit the enzymes on the surface of the lymphoma cells and interrupt the downstream signaling that the cell requires for its growth and survival, as well as idelalisib, another kinase, another enzyme inhibitor, and certainly this agent is currently being submitted to the Therapeutic Goods Authority for their approval on the basis of the clinical research that has been led by Professor Sahls and other of his international colleagues into this new enzyme inhibitor. And lenalidomide, the sister agent to thalidomide, which sends shivers in everyone's spine, is actually a very powerful immune modulatory agent and can, educate our own immune system to try and control the lymphoma. And immune checkpoint blockade, the nivolumab, how do you pronounce it? I've still got to get my tongue around so many of these. Nivolumab, all right. Thank you all. and pembrolizumab, whatever. They're all very exciting blockades of the programmed cell death pathway. So, not every patient is appropriate for a clinical trial. And we've all heard about the enormous spectrum of the different types of lymphomas. And so, it's very important. No one clinical research unit can possibly have a broad enough clinical trial portfolio to be able to offer to every patient with lymphoma with newly diagnosed or relapsed disease. And I've borrowed this slide from one of my French colleagues, and it emphasises to me the importance of the individualised therapeutic decision-making that needs to go on when one considers a patient with relapsed lymphoma. We have to consider the subtype of the lymphoma, the location of the lymphoma, the patient's own age and fitness and their own willingness to have treatment, in addition to the previous treatment that they have received. But today's physician is not a cantankerous Dr. House who is the autocrat who says you will do this with astounding brilliance. Today's physician is a team, and multidisciplinary team meetings are being held all around Australia on a very regular basis with clear documentation, discussion and debate around the diagnosis, the staging, the previous treatment, and what are the appropriate therapeutic approaches for patients with relapsed lymphoma, and whether there is a clinical trial that they should be screened to see whether it is appropriate to offer the patient participation in that trial, to talk to them about the purpose of the trial, the procedures involved in participation, the potential benefits and the known risks of participation, along with the alternative treatments that they would be offered if they weren't participating in that trial, and importantly stressing the fact that clinical trial participation is a voluntary act by yourself, not just the moment you sign the consent form, but during the process of that entire years and years of participation in the trial. And I assure you it's not years and years usually of treatment, but years and years of follow-up in a trial. So, clinical research is integrating our clinical expertise with the best available external evidence from systematic research. And it's very important to point out that that is actually a bit different to just expert opinion that we may find on the net or elsewhere. And it's very essential that we bridge this divide between the sciences that underpin our knowledge of our disease and the application of that science in clinical practice. So what is a good clinical trial? Well, firstly, it's one that asks the right question. And that might not be the question that Professor Rimmel or Professor Sala or I think of, but the question that we think of on your behalf. It's got to be reflecting the community's values and the community's priorities. It's got to be a trial that you want to participate in. It's got to be ethically justifiable and conducted ethically, and we have a very, very rigorous code of good clinical practice that is applied internationally. And the question has to be able to be reliably answered. The statistics have to be right so that we can actually get an answer at the end of the trial with minimal bias. And so a clinical trial is a carefully designed study conducted under the code of GCP that seeks to determine under controlled conditions the safety and effectiveness of a new treatment method or a drug. And it's this that allows those laboratory breakthroughs in the test tube to actually be converted into our clinical practice and become new standard of care for treatment. So I'm going to go through with you the drug development timeline. When, on the basis of a biologic, a scientific rationale, a new agent is tested against various cell lines, and then after animal testing, that actual process takes a good 2 to 4 years. The actual clinical research and development of a drug can take 3 years to 10 years. And that's because the first clinical trials in humans have to be all about safety. Can we, in 20-odd patients, deliver increasingly small doses, increasingly larger doses of this agent without causing undesirable, intolerable side effects in humans? And after that phase 1, we move on to phase 2. Does this drug, whose toxicity profile we know, whose dose we've tried to work out on the basis of the phase 1 testing is the best dose to be giving, does it actually work? Does it actually have efficacy against lymphoma in patients? And these phase 2 trials may be just 100-odd patients. And then come the very expensive trials to conduct that Professor Saal mentioned, and that is the phase 3, the randomised clinical trial, where we are comparing the new treatment approach with the standard treatment approach that's in current practice. And we acknowledge that many of these phase 3 trials, if not most of them, are industry-sponsored studies because of the sheer cost of conducting under the code of good clinical practice, the very expensive, there's huge numbers of patients required for these trials, but we do assure you we work very hard in partnership with industry and also on our own, in our own collaborative groups around the world in collaboration, country to country, to try and to design the randomised controlled trials that answer the questions that are important to our lymphoma patients. A new drug application is something that we could end up spending and debating for the next hour or so. We acknowledge that in Australia this is a very long process, but it is also a very robust process to ensure that the Therapeutic Goods Authority only approves agents that are demonstrated to be safe and effective, and the Pharmaceutical Benefit Scheme only funds agents that the Australian pocket can afford. And then the other important thing that mustn't be forgotten is the importance of post-market surveillance, so that we don't end up with another thalidomide scare, another Vioxx scare, because clinical trials, even if they're done in thousands of patients, do not provide the full safety and efficacy profile of any new agent. So why would you yourself participate in a clinical trial? And the obvious first answer is the direct benefit. But that's the direct benefit not just from the studied therapy, but even when the treatment is standard, research has demonstrated that patient outcomes are better. And that's probably in part due to the very rigorous monitoring and follow-up that patients get within their clinical trial. All my clinical trial patients love the fact that they have one-on-one care coordination with the clinical trials coordinator who knows where to track me down when the counts are low, they've got a bit of nausea, when they need instant medical advice. There's also the benefit to medicine and to science, and the benefit to future patients. We are all by nature altruistic and having benefited from the patients who have participated in the clinical trials before us, we want to make our own contribution to clinical research. It speeds development of effective therapies, but importantly also clinical trials research stops the development of ineffective or overly toxic therapies. And also here, particularly a particular motivator for us here in Australia is it does offer us early access to therapies that are not available on the Pharmaceutical Benefit Scheme. Even though bendamustine is not yet PBS approved, PBS subsidised in Australia, I know there are a number of you who have accessed this agent in the context of clinical trials when pairing bendamustine with other therapies. So why not participate in clinical trials? And there are a number of very right reasons for not participating. And the first one is the particular trial that you're being offered might not be answering the right question, might not be giving what you think is the right therapy for you. And I urge you, if you have your hesitations about trial participation, to express them very loudly and clearly to the physician who's offering you participation in that study. The direct risk of the unknown nature and the limited knowledge that we have about agents, particularly in phase 1 and phase 2 testing, must be acknowledged. And it's certainly a hell of a lot easier to just have the standard of care that your doctor is going to prescribe for you. And for many patients, particularly those when their lymphoma is newly diagnosed, to consider participating in making a decision about participating in a clinical trial, reading the 20-page document that was 8 pages when we first drafted it and then became 20 pages after lawyers got to it, it's just too much for patients to absorb and consider, particularly patients for whom English is not their first language. Clinical trials participation is a little bit more onerous. It's more onerous for the patient, it's certainly more onerous for the clinician, because there are more tests and more visits generally that happen, probably by the order of at least another 10%, because of the very close monitoring that is required usually in clinical trial protocols. And it's important to ask always, when we consider whether we want to participate in a trial, and you also should consider, who benefits from participation in this trial, and also the implications of whether, if I was on a randomised trial and I didn't get access to what I think might be the new Butte next thing since sliced bread, is it going to be possible for me to get access to this agent in the future? And I think it's very important that we're always mindful about the competing priorities that we have in terms of both patients and industry in the conduct of clinical trials. But nonetheless, I think it's very important that we have to counter the negative perceptions of trials that exists out there in the community, where people who participate in clinical trials are perceived as human guinea pigs, because public awareness of clinical trials is not the same as public understanding of them. Now, in New South Wales, but also in every state in Australia now, except for Queensland, but the Queensland app is coming, is a mobile app that helps us busy clinicians keep tabs of all the currently conducting, the currently recruiting clinical trials that are available locally. All right, we have at least half a dozen haematology apps and many other apps for other cancers. And this app is, and it's not something that I necessarily encourage you to download, but I want you to be aware that it's in your clinician's pocket almost certainly. And they are keeping themselves briefed as to what are the new agents that are currently being tested in clinical trials that are currently recruiting in Australia. And since we launched this app 20 months ago, there have been an average of 10 patients a month who have been referred to other hospitals in New South Wales to participate in clinical trials. Because ultimately, in Australia, we do have that, all physicians want to be able to be offering to patients what they feel is the best therapy for them, and physicians increasingly have no compunction to recommend to their patient that they go elsewhere, whether it be Royal Prince Alfred Hospital for someone who's at Concord, whether it be Royal North Shore Hospital or Westmead, somewhere that is feasible for the patient to commute to participate in clinical trials. Notwithstanding that, we have had many patients from Milton in southern New South Wales, from Dorrigo in northern New South Wales, from the Central Coast. Patients will travel quite long distances to access new therapies in clinical trials. So, in Hodgkin lymphoma, I just want to comment on one new agent that has occurred and become increasingly available, not so much in clinical trials now, but because it's, as Professor Saul pointed out, this anti-CD20 drug conjugate with the toxin has been proven to be so effective, it's now actually been studied in first line. And this anti-CD30 antibody, Adcetris or brentuximab, is very effective in treating patients with relapsed Hodgkin's lymphoma, either after they've had a transplant for their first relapse or after they've had a second relapse. And there are excellent response rates. Generally, about 3/4 of patients will have a response and 1/3 of them will have a complete response. But the main side effect that occurs after weeks and weeks of treatment is this numbness that requires dose adjustments and dose delays. The bottom line is it is extremely expensive, and this is one of the reasons why, although it's approved by the Therapeutic Goods Authority, it is not yet PBS listed here in Australia. It is also another agent, as I said, that is being used and is approved and PBS listed for certain patients with relapsed T-cell lymphomas. And in the interest of time, I'm not going to go through the PD-1 inhibitors, but certainly they are available in clinical trials for patients with relapsed Hodgkin lymphoma in Australia. I think I'm going to leave any discussion about low-grade lymphoma to the breakout session that we're going to be having here, because that is going to be webcast. And there is a mind-boggling plethora of agents that are currently in clinical trials for patients, both with relapsed and newly diagnosed follicular and other low-grade lymphomas. I might say here, perhaps if any of you have marginal lymphoma, zone lymphoma, I think that's a very unfortunate title, because I'm increasingly feeling that marginal zone lymphoma patients are very marginalised, and it's very difficult to find clinical trials that they can participate in, and that has implications potentially for their access to these agents when they're approved. For relapsed aggressive lymphomas, there aren't so many trials recruiting currently, but I know I looked up the app today, and there's one called Checkmate, which is a nivolumab study for patients with relapsed diffuse large B-cell lymphoma. Clinical trial is certainly, I strongly believe, the best therapeutic option for patients with relapsed T-cell lymphomas. They are very difficult diseases to get back into remission and keep in a sustained remission after these lymphomas have relapsed. However, large clinical trials for T-cell lymphomas are not very commonly available. We've just had one close, and we'll have another one open soon here in New South Wales, using the selective inhibitors of nuclear export, i.e. just another targeted therapy for lymphoma that has significant promise. Basically, clinical trials are really designed to help you live longer and live better. And through clinical trials research, there is no doubt that hundreds of lymphoma patients in Australia have had access to all these agents, but importantly also to personal care coordination by their study coordinator. After relapse, it's very important that everyone keeps hope, but also to acknowledge that those hopes sometimes change. And it's important that every patient, maintains their hopes, works out their milestones, because I think as a physician, when talking to patients about participating in a clinical trial, we have a very delicate balance between not overselling the trial, and we need to foster realistic hopes for remission. We need to be very careful that we're not peddling false expectations. But nonetheless, for me, the joy of lymphoma care certainly comes from the rapid advances from bringing research into clinical practice, and also how often after relapse my poor prognosis patients prove me wrong through their participation in clinical research. Thank you all very much indeed.